Okay, this video is for the recombinant DNA activity, which is labeled the E. coli insulin factory. What you're going to need are the directions. You'll need a roll of tape. You'll need some scissors. Okay, and then ideally, if you had different colored pieces of paper, they're labeled. This one says print on green paper. It's fine. This one if it's on white. Same thing with this one. It says blue, but if you print on white, no problem. If you do have any colored paper, it would just be nice to be able to differentiate the plasmid DNA from the cell DNA. So those would be perfect if you could do that. But anyways, I'm going to cut these up now, as well as my restriction enzymes, and then I will be right back. Okay, so what you have now are 8 restriction enzymes and ligase, which, as you remember, is the enzyme that is going to join the sticky ends together. We think of it as like a glue stick. You have your DNA that is going to have your human insulin gene, and then this is going to be used for your plasmid. Now, one thing, notice before you throw everything away, this little key here that you're going to need at the end, that's on the pink paper or the plasmid DNA paper, so I don't want to throw that little bit away. Okay, so first what you're going to do is you're going to build your DNA. That's going to be for your human insulin. So these ones do need to be in a specific order. So you're going to match up the numbers. Here I have a 1 at the bottom and a 1. So those are going to match up. Okay, and you're just going to tape it together. Okay, it doesn't have to be pretty. Okay, then the 2's match up. Now this is supposed to be human DNA, so it's going to be linear rather than circular. Now as we build this, notice that this part here is shaded. The shaded part represents the DNA that's actually going to code for insulin. So that's going to be an important piece. It's a little bit unwieldy. As you can see here, now I have built my human DNA. Okay, and this again is our target sequence that codes for insulin. Now I'm going to build my plasmid DNA, and the idea here is that every group might have a different plasmid. So the instructions tell you choose the strip that has the origin of replication, and then 3 other random ones, and we're just going to randomly attach these together so that everybody's is different, or most likely. Okay, I'll just choose the first 3 that I've randomly set up. Two, 3, and then this is plasmid DNA, so we know that plasmid DNA is circular. So once you have your 4 strips, you're going to tape them together, and you've now made your plasmid DNA. These 2 can be recycled. Okay, so set this aside for now. You're going to need it at the end. So we have plasmid DNA and human DNA. Our goal in this activity is to find an enzyme that cuts out insulin. So you're going to want to have it cut above the insulin gene and below it. Whoops. And then also cut 1 time on the plasmid so that we can use the same restriction enzyme to cut out the insulin gene and then paste it into the plasmid. Think about the consequences here. If you have it cut twice in the plasmid, that's not going to work because you're going to have extra fragments of plasmid floating around. And if it cuts, say, in the middle of the insulin gene, that's not going to work. So you'll have to reject it because you wouldn't have the insulin gene if you cut it somewhere in the middle. If there are 2 different enzymes that work, if they cut somewhere above and somewhere below this shaded insulin gene, say there's 2 different options that work, you're going to want to cut it as close as possible. So choose the one that cuts closer to where the insulin gene is as your ideal enzyme. So I'm going to set ligase aside for now. And let's go ahead and just start with 1. And what you're going to do is indicate anytime you see this. So however you find the easiest, we're gonna go through CTT. So I'm just gonna look for CTT. I'm gonna go through this whole thing here, CTT, AAG. Okay, here's a match. So you're just gonna draw on here where the enzyme would cut. Remember, the enzymes aren't gonna cut straight across, right? They're gonna cut like this, leaving sticky ends, and that's gonna be really important. Be sure you label it so you know which one it is. If it cuts like this, then when you insert it into the plasmid, if you use the same enzyme, the sticky ends are going to base pair with each other, and so it's gonna fit in to wherever you use this enzyme to cut the plasmid. So I'm just gonna keep going. I'm looking for CTT, CTT, here's CTT. Okay, good. Whew, if that had been in here, we would have had to throw out this enzyme because we have to make sure that it doesn't cut our insulin gene. CTT, nope. CTT-AAG. So here's another location where it will cut. So I'm going to draw this one in. Okay, I'm going to label it eco. Okay, let's see if there's any more. CTT. CT, nope, it has to be exactly the same, so that doesn't count. Okay, so I went through all of the locations on the human gene. I'm gonna now go through the plasmid. If it's circular, so it's easy to get kind of messed up. Consider starting at the origin of replication so that you know when you get back to it, you've gone all the way around. So I'm looking for CTT. Oh, that's so close. CTT-AGG, but nope. Not gonna work. CTT, oh, here's one. CTT-AAG. So I'm gonna, okay, I'm going to put that one in. See if there's any more. If there's another one, I reject it because it can only cut once, and there's not. So let's set this aside for now. This one's a good possibility because it cuts one time on my plasmid and on either side of my insulin on here. So what I'm going to do now is I'm going to go through all 8 of these and do that same process. So you can look for GGCC. GGCC, here's one right here. Okay, so I'm going to go through here. Okay, be sure you label it. Okay, GGCC. GGCC, HPA2. Anyway, okay, so you can go through this. If there's any in the middle here, it won't work. Anyways, okay, so what you're gonna do is you're gonna then, once you've gone through and labeled all 8 of the restriction enzymes, you're gonna find the one that, as close as possible, okay, cuts out your shaded insulin gene without cutting the middle of the gene. And remember, it has to also cut the plasmid once. So I haven't gone through and done all 8, but I'm just gonna show an example. Say that one, and remember, everyone's plasmid is different. So you might have a different restriction enzyme that works best for your particular setup. So I'm gonna say that I went through all 8, and my ideal one is this first one that I looked at. So what you're gonna do then is you're gonna cut out along your line. Okay, so then I'm gonna say I'm using this plasmid. Okay, it cut here once, and it cut here once. Cut here. Okay, so this is genetic modification. Now, so I can use, where did I put my ligase? Here's my ligase. Okay, attaching my sticky ends. And notice that all of the sticky ends, because I used the same restriction enzyme here, are gonna base pair accordingly. Okay, and I'm gonna use my ligase. Okay, and I'm gonna just attach that in here. And now what I've made is I've taken my plasmid. You can, I don't know, this is kind of big, but it's all circular now. I've inserted my gene that codes for human insulin into my plasmid. I can then insert this into a bacteria or a yeast that can then make human insulin for me. So this is what I'm going to turn in, but there's also an extension. So you need to fill out everything on this form for each of these 8 enzymes, how many cuts it did on each of these, which one you used, reasons for use or non-use. But then the last portion is where this piece of paper is going to come back into play as being important. So on your plasmid, remember everyone's plasmid is different. You may have areas that code for certain types of antibiotic resistance. So you're going to need to go through, take a look at your plasmid, map it out, and indicate which antibiotic resistances your plasmid contains. Now, all of the instructions I just went through are in this packet. So if there was anything that needed clarification, read through this carefully. On the back here is where it talks about antibiotic resistance. So yeah, hopefully that works out. Good luck.